So, much of science uses something called fluorophores for light microscopy. A fluorophore is a compound that absorbs light, it absorbs your excitation light, and then it'll emit light back out, and that is called your emission light. So, you can tag certain proteins with fluorophores via something called antibodies. So, when you shine a specific wavelength of light onto these fluorophores, they can shine back at you and you can image them. And since these fluorophores are attached to certain proteins, you can look at your protein of interest. This is called immunofluorescent imaging. So there are many types of light microscopy. Two types that I'm going to be talking about is Widefield and Confocal microscopy. So, Widefield microscopy is one of the more basic versions of light microscopy. The most basic version, I think, is just using your transmitted light to image your cells. This is the most basic because there's no specificity. All you're going to see is the overall shape of your cell. So, wide field microscopes use LEDs to excite your fluorophores, and it does so by lighting up your entire sample at one time. So, this basically means that the fluorophores on pretty much all of the focal planes in your sample are going to light up at once. This also means that when you take your image with wide field microscopy, your image will be brighter but blurrier, aka you will have less resolution. For those of you who don't know what resolution is your ability to distinguish two things from each other. So think about like a lawn, right? You have a really big lawn and you have a really shitty camera. You take a picture of the lawn and it's a little blurry. You can't really make out the different blades of grass because your camera is shitty. But if you have a really nice camera and you take a picture, then you can see each individual blades of grass and you can distinguish them from one another. That is resolution. Having higher resolution is being able to see the individual blades of grass. So, wide field microscopy doesn't have amazing resolution, but confocal microscopy increases resolution. So, confocal microscopy uses lasers for the excitation light instead of LEDs. So basically, the laser light will enter through something called a pinhole. This focuses all of your light down to a singular focal plane. So before in wide field microscopy, you would light up every single focal plane at one time. For confocal, you will just light up the fluorophores in the focal plane that you want. This is why you will have better resolution. Then, when the emission light, again, you have your excitation light, which is the laser, the fluorophores will shine back emission light. This emission light will enter the camera, and it also goes through a pinhole when entering the camera, so it gives you only the light from your focused focal plane. So basically, confocal microscopy gives you axial selectivity, meaning that only the fluorophores you want to see will be excited, which again means more resolution, which is the name of the game. There are specific reasons why you would want to use confocal microscopy, mostly if you want to see smaller structures. For example, I do a lot of imaging of DNA damage. This appears in the form of foci. So you would want to quantify the number of foci within your sample. And the best way to do that is by having high resolution, just in case you have two dots that are close to each other and you can't exactly distinguish them. It's for these types of things that you would want to use confocal microscopy. I will insert a few pictures so then we can all look at the difference between wide field and confocal microscopy. Probably I will insert pictures of the protein of interest I'm imaging right now, which is lamin AC and lamin B1. Also on the left, we can see that it is of a wide field image. You can see how the entire nucleus is lit up because again, all of the fluorophores in my sample are being lit up at once. However, on the right side, we say a picture of a confocal image of the exact same nucleus. Okay, maybe not the exact same, but they're from the same sample. You can see that there is a very nice localization of lamin AC around the periphery. This is because in confocal, I chose that one specific focal plane to light up those fluorophores. Again, you wouldn't want to use confocal microscopy for something like this because I want to see specifically how much of this specific lamin AC will localize around the periphery. And I soon will probably talk about something called a super resolution microscopy. Again, you have like tier lists of different versions of microscopy. It goes wide field, confocal, super resolution, maybe ESM, electron scanning, I think, microscopy. And I've only done one version of super resolution microscopy that's called STED imaging. I will insert a picture of STED imaging here. So on the left side we have confocal, and on the right side we have the exact same image, but with STED super resolution microscopy. And you can see that you are getting way better resolution. I feel like I'm getting around 100 nanometer resolution on the STED imaging. If you look close, you can see a lot more detail within my sample. Again, this is due to better resolution. Resolution is the name of the game if you want to see very, very small structures. Did I already run out of things to talk about? Okay, let me finish this really quick. And I'll put in other images, other example images of confocal versus wide field microscopy, just so we have a nice little slideshow to look at. Well, I finished typing this. But I suppose I can talk about AI versus human art. I think we should be afraid of AI, generative AI specifically, especially if you're a fan of the arts. I'm a big writer, and along with the internet screwing up people's attention spans, no one's going to want to read a book. No one wants to sit down and write a book, you know? So people will use AI, people will use Spark Notes to get through their book. I was just talking to a friend of mine, and he said the last time I read a book was in 8th grade, which at that point is like 6 years ago, which is absolutely crazy to me. But hey, I've fallen victim. I haven't read a book in a while too. And I'm afraid of AI, you know? I'm afraid that we're going to get to the point where you can't distinguish AI versus human art, decreasing the resolution maybe of our perception of AI. Anyway, let's read what I wrote, shall we? Confocal. I'm two. Through Easter, then Monday, then the night. I'm you. On stage in the dish under the light. I'm each blade of grass in a landlocked field, from 7 miles high in the window seat, and the dirt in between the feet with mud, worms, and fertilizer and meat. If I'm known, I'm not seen, indistinguishable from the other version of me. I have a deeply rooted madness that even the light, God's light, cannot see. Raise me to confocal, otherwise I'll seem like one, a body of Easter and Monday in the night. I'm likely to be everything. Afraid of resolution, afraid to be none. I like it when poems rhyme. You know, I don't know if there's even a rhyme scheme here, but I like it when a good poem rhymes. I also don't really read a lot of poetry, so I'm really not practicing what I preach, so it makes me a hypocrite. But just let's not use generative AI. It's just not good for you. It's not good for your psyche, and it's not good for your soul. I'm not a huge fan of this poem I wrote, but at least it incorporates confocal microscopy, which I think is the most important lesson that we should take away from this.